Lecture 5, Life History, Learning from the Environments of Others. Student learning objectives. Students should be able to discuss how the degree or level of predation may impact the prey population's life history in terms of these 4 strategies: age at maturity, size of offspring, number of offspring, lifespan of population. Introduction. Theories of life history are concerned with explaining the whens, whys, and hows of key events in an organism's life. These events include, but are not limited to, the 7 areas listed below. We shall, in this lecture, however, focus on the first 4. 1. Age at sexual maturity. 2. Size of offspring. 3. Number of offspring. 4. Lifespan of population. 5. Parental investment in offspring. 6. Senescence. 7. Death. In this lecture, using the interactions between guppies and their fish predators in the northern rivulets of Trinidad as a model, we shall attempt to investigate some of the life history strategies as they play out in the relationship between predator and prey, killifish, pike cichlid, and guppy, respectively. In the figure below are a pair of guppies. The female is to the left of the male. Males are generally smaller than females. Males are also more colorful, with larger and flashier caudal and dorsal fins. The difference in appearance between males and females in the same population is called sexual dimorphism. In the next slide, comparative anatomy is used to explore the dimorphic differences between male and female guppies. Comparative anatomy, guppy dimorphism. A male guppy is depicted in the figure above, a female in the figure below right. The major male anatomical parts are, from left to right, pectoral fin, dorsal fin, caudal fin, gonopodium, anal fin, ventral fin, and the operculum. In the female diagram below, right, the anatomical parts are the same except the anal fin is without a gonopodium, and the female has a gravit spot and a vent. In summary, the 5 characters that most sharply distinguish males from females are the following. 1. Males have a gonopodium, females do not. 2. Males have larger dorsal and caudal fins than females. 3. Males are smaller than females. 4. Females have a vent, males do not. And 5. Females have a gravit spot, males do not. Note that in nature, there is often a trade-off between survival and sex. Thus, the very large and flashy dorsal and caudal fins of a male left may be more a result of artificial selection by breeders trying to attract human customers into pet shops than of males trying to attract guppy females. For in nature, larger, bright fins have and will attract predators as well as females. That is, predators put pressure on male guppies to be less conspicuous. The effect of the pike cichlid on guppy color is depicted next. Predator coarse gravel experiment. The initial guppy population depicted in figure A is living with the predator in a coarse gravel environment. That guppy population evolves over 15 generations to fit in with its environment, as depicted in figure B. That is, large spotted guppies become more populous, being eaten proportionately less frequently, so they produce more of their kind. The predator in a fine gravel experiment. The initial guppy population, as depicted in figure C, is living with a predator in fine gravel, and it evolves, that is, the prey evolves over 15 generations to fit in with its environment. D represents the small spotted guppies that evolve and become more populous, being eaten less frequently than the larger spotted guppy. So the smaller spotted guppy produce more of their kind. In the following diagrams, there is no predator. In A, the initial guppy population is living without predators in coarse gravel. In figure B, the population evolves over 15 generations to contrast sharply against the coarse gravel environment, because the male guppies with smaller spots are easier for females to select for sex. No predator in the fine gravel experiment. Figure C depicts the initial guppy population living without predators in fine gravel. Figure D represents the same population after 15 generations of living without the predator in the dark environment. Notice that the spots have become larger over the 15 generations, and they sharply contrast with the fine gravel environment. The large spotted guppies have become more frequent in the population because the large spots are easier for females to select for sex. Here we summarize the findings. 1. When predator is present, that occurs in A1, B1, A2, and B2. Guppies live longer if they blend into background. If they live longer, they reproduce more guppies. So the strategy here is best blenders equal best reproducers. In the second set of diagrams, C1, C2, D1, and D2, guppies contrast with their environment, so they meet more females and reproduce more guppies. Most conspicuous equals best reproducers. The take-home point for males, the trade-off is between living longer and reproducing more. Life means choosing between more offspring and more life. More offspring means more guppies that look like you, but you, in fact, will have a shorter life. For to have more guppies, you have to be seen by females. But to be seen by females is also to be seen by predators. Point of contrast. Guppies living under high predation, when moved to low predation environments, do better than low predation guppies in low predation environments. In contrast, in some plants, it is possible that the plants in their own native environment do better than plants that are newly introduced into non-native environments. Thus, the experience of one group of organisms does not necessarily mean that a similar experience will occur for a distinct group of organisms. Returning to our guppies, using greater detail, we further reinforce our previous findings. Slide 12, in the top diagram, there are 3 pools of water. Proceeding clockwise from upper left, the pools are numbered 1, 2, and 3. Pool 1, a control pool, has guppies and killifish who feed on the small guppies. Pool 2, experimental pool, also has killifish, but initially there are no guppies. And pool 3, also a control pool, has both guppies and a pike cichlid who feeds on the large guppies. Pools 1 and 3 serve as comparison pools. Pool 2 is the pool where the descendants of guppies from pool 3 are transplanted. Pool 2 is the experimental pool. The purpose of the 11-year experiment was to determine the impact of pike cichlid predatory pressure on guppy life histories. 200 guppies in the first year of the experiment moved from pool 3 to pool 2. The weight and age at maturity of the pool 3 and pool 2 guppies were compared to determine the impact of pike cichlid predation. The results of the 11-year experiment are reported in the bar graphs at the bottom of the slide. To read the 2 bar graphs, we need 3 pieces of information from each bar graph. We need to know what the vertical axis measures, what the horizontal axis measures, and what the 2 colors, blue and purple, mean. The vertical axis measures a life history attribute, either mass at maturity or age at maturity. The horizontal axis distinguishes between males and females. And the blue color says that the guppies are living in pool 3 with the pike cichlid, the control group. The purple says that the guppies are living in pool 2 with the killifish, the experimental pool. Let us read the bar graph on the left. The male guppies living with killifish, purple bar, are larger at maturity, 76.1 milligrams, than the male guppies living with the pike cichlid, blue bar, measuring 67.5 milligrams. And the female guppies living with killifish, purple bar, are larger at maturity, 185.6 milligrams, than the female guppies living with the pike cichlid, blue bar, measuring 161.5 milligrams. The findings are similar when the average age of guppies living with the killifish, purple, are compared to the average age of the guppies living with the pike cichlid, blue, as is demonstrated by the bar graph on the right, where the vertical axis measures the age in days of guppies at maturity. When guppies are moved from pike cichlid pools, blue, to killifish pools, purple, their average age at maturity increases for both sexes. Male age at maturity increases from 48.5 days, blue bar, to 58.2 days, purple bar. And the females increase from 85.7, blue bar, to 92.3 days, purple bar. The conclusion in both cases is that guppies living with pike cichlid, blue bars, weigh less and are younger at maturity than guppies living with killifish, purple bars. In this experiment, guppies are studied to determine the impact of predation on guppy color. Guppies initially lived in 3 environments, which are represented by the 3 columns of the top row. High predation, 1. Guppies are living with pike cichlid. 2. Low predation. Guppies are living with killifish. And 3. No predation. Guppies are living alone. The results of the experiments are displayed in the bottom row. In the 1' high predation area, the pike cichlid eats bright colored fish. So the dark fish live longer and produce more offspring because they are able to hide from the pike cichlid. Therefore, the proportion of light fish declines from 4 out of 8 to 2 out of 8. Thus, predator selection has the largest impact on male guppy color. In the 2' low predation environment, Rivulus eats the light-colored guppies, but Rivulus eats so few that the proportion of light-colored guppies increases from 4 out of 8 to 6 out of 8 because the females prefer light-colored males. Sexual selection has the largest impact on male color. In the 3' no predation area, the number of colored male guppies increases over time from 4 out of 8 to 7 out of 8, following a color pattern similar to that of the low predation area, and thereby reinforcing the finding that predators affect guppy populations by reducing the color intensity of the males. The lack of any predator pressure on the male population results in only female sexual selection pressure pushing males to brighten up. In this experiment is measured the impact of predation on guppy color again. There are 3 bodies of water, 1, 2, and 3. Body of water 1 is the Rivulus only tributary lacking in any guppies. Body of water 2 is a high predation region of the Aripo River with pike cichlid and other predators. And body of water 3 is a low predation area with guppies and ribulus or killifish. 200 guppies are transferred from the high predation area into the river that only has the killifish. After 2 years or 15 guppy generations, the transplanted guppies are as colorful as those in the low predation tributary. This experiment reinforces the findings that high predation environments impact the color of male guppies. Life history strategies. Organisms allocate their resources to three broad areas: self, offspring, community. The amount of resources allocated to self, reproduction, and community, and the timing of the allocations to each is a life history strategy. We shall be looking at a modified conventional four allocation life history model, to which one additional allocation, community, has been made. The conventional model identifies four energy allocation areas. The first four in the list below: 1, maintenance or upkeep; 2, growth; 3, storage; 4, reproduction. The fifth area is controversial: 5, community. We can collapse the four allocation model into a two allocation model: self and reproduction. In the animal world, the big trade-off is between surviving and reproduction. Surviving requires finding food. Finding food usually involves exposure to predators. Mating often requires double exposure to predators, once for food to survive and once for a mate. Surviving can be reinflated into maintenance and growth and storage. And maintenance can be further expanded into repair and storage and defense. The relative importance of allocations vary by populations and species. On the next slide is a diagram comparing the energy allocations of snakes and humans. Snakes are ectotherms. They obtain their heat from environmental sources external to their own bodies. For example, the sun. Humans are endotherms. They obtain their heat from metabolic sources internal to their own bodies. For example, sweating. Metabolism is a set of life-sustaining chemical activities involving biomolecules within the cells of living organisms. Metabolism is a set of life-sustaining chemical activities involving biomolecules within the cells of living organisms. For 100 units of food, snakes' first use is 50% of the food energy for growth. Humans' first use is 50% for maintenance or upkeep. Snakes' second use is 16% for reproduction. Humans' second use of food is for waste elimination. We look more closely at maintenance. Maintenance upkeep is the first energy allocation area. It is also called homeostasis. It is the organism's allocation of energy and other resources to keeping itself alive and in good health. Maintenance operates at the level of the cell, the tissue, the organ, and the whole organism. regulation of temperature, endotherms, mammals and birds maintain a constant temperature. Regulation of blood pH. For humans at about 7.4, usually slightly basic. Regulation of blood glucose level. Insulin is a hormone secreted by the pancreas that regulates blood glucose sugar. Without insulin regulation of blood glucose levels, diabetes occurs. 2. Growth is the second energy allocation area. Cells, tissues, organs, and whole organisms need energy to grow larger and to grow more complex. The organism must allocate energy and other resources to growth activities. Storage is the third energy allocation area. Organisms save energy and resources for hard times, rainy days, and quick fixes. Offspring reproduction and protection is the fourth energy allocation area. Energy and resources are used to produce or protect offspring. Parental investment measures the energy and other resources allocated to creating and sustaining offspring. Community is the fifth energy allocation area. Community is not applicable to all populations. Community is well defined in Hymenoptera, that is, bees, ants, wasps, and termites, and possibly naked mole rats. This is a naked mole rat. Notice that there are no visible eyes and no hair. Life history strategies. Life history strategies can be seen as a type of optimization strategy of microactivities at the individual organism level. A life history strategy is performed by each organism separately, but the similarity in the strategy appears as a pattern across multiple individual organisms in a population. Thus, similar behavior of individual members within a population is so consistent that it is possible sometimes to compare the behavior of two populations on the basis of the within population similarities. So what do we mean by within a population and between populations? Comparison within a population means that we take a population of the same type of organism as a population of squirrels living in a yard with many trees and no predators, and we compare the behavior of the individual members of the population to each other. Comparison between populations, we compare the population of squirrels living in one yard to the population of squirrels living in a neighbor's yard. Now we are not looking at how the squirrels in one yard differ from each other, but rather how the squirrels in one yard differ from the squirrels in another yard. If all the squirrels in yard A get up at the same time every morning, say 6 a.m., to search for food, and all the squirrels in yard B search at 2 p.m., this is a between population difference in the two populations. Life history strategies in this lecture pertain to the guppy, to the guppy's size at birth, age at maturity, allocation to reproduction, how many, how large, how often. We do not look at area 4 particularly, but area 4 is senescence and menopause, which for the guppy does not exist. That is, menopause does not exist. Life history changes as a function of environment. Life history, therefore, can be seen as an adaptation or series of adaptations to pressures from the environment. For example, a change in environment from low to high predation impacts the life history of guppies. Size and age and maturity, size and number of offspring, and life expectancy. The guppies adapt to the pressures that predation places upon them. Now we shall think about a life history model for guppies facing predation. Standard life history theory predicts that high mortality among adults will lead to, that is, select for, early maturity and increased reproductive efforts relative to growth, storage, and maintenance allocations. Why? Answer, because in high predation areas, early to plate results in early to mate. Suppose guppies vary in age at time of reproduction between those that reproduce at 10 months and those that reproduce at 16 months. Since growth for female guppies continues throughout their lives, then at 16 months, females will be larger than at 10 months. Assume further that older, larger females produce healthier, larger offspring. Also assume that larger female guppies are more likely to be eaten by pike cichlids. So in the pike cichlid pond, larger guppies are more likely to be eaten than smaller guppies. And if they mature late, then they are likely to be eaten before they reproduce. If they are more likely to be eaten before they reproduce than guppies that mature earlier, then over time, the relative number of genes for early maturing guppies will increase in the gene pool, while the relative number of genes for late maturing guppies will decrease. As a consequence, the frequency of guppies who reproduce at 10 months will increase relative to the guppies who reproduce at 16 months. So the guppy population will be smaller and reproduce earlier. Take-home points. 1. Predator pressure on prey communities affects prey life histories. 2. In the case of guppies, 6 effects have been identified. 1. Females under high predation pressure mature earlier than females under low predation pressure. Females under high predation pressure are smaller at maturation than females under low predation pressure. 3. Females under high predation pressure produce more offspring than females under low predation pressure. 4. Females under high predation pressure produce smaller offspring than females under low predation pressure. 5. Females under high predation pressure have shorter lifespans than females under low predation pressure. And finally, 6. Males under high predation pressure are less colorful, duller than low predation pressure males. Can you specify additional life history strategies that male guppies employ? Take-home points continue. 3. Understanding a life history of a population includes knowing its predators and prey. Environmental pressures on an organism, including predatory and sexual pressures, sometimes work together, but quite often drive organisms in different directions, creating a trade-off between survival and sexual reproduction. And so, to understand the significance of a phenotype, the appearance and behavior of an organism, it is necessary, in addition to genetic components of the phenotype, to determine the underlying environmental influences that produce the phenotype. What are the predatory pressures? What are the sexual selection pressures? Are these pressures complementary or antagonistic, pushing the population in the same or different evolutionary directions, respectively? Problem set, Lecture 5. This is the end of Lecture 5. Thank you.